In this short presentation, we'll look at how photorespiration undoes the work of photosynthesis and how some groups of plants avoid it. In the previous presentation, we ended up by looking at the Calvin cycle. In the Calvin cycle, the carbon from carbon dioxide is bonded to the 5-carbon molecule RUBP, ribulose bisphosphate, to form a 6-carbon molecule. The enzyme responsible for this reaction is called Rubisco. Now, under ideal circumstances, Rubisco binds carbon dioxide to bond it to the RUBP. However, if there's a lot of oxygen present, Rubisco will bind to the oxygen instead of the carbon dioxide. This process of Rubisco binding oxygen starts a process called photorespiration. So again, under normal conditions, Rubisco binds carbon dioxide to RUBP to form 3-phosphoglycerate, which is used in the Calvin cycle. When there's a lot of oxygen, however, photorespiration may happen. In photorespiration, Rubisco combines with oxygen to form a molecule of 3-phosphoglycerate that's used in the Calvin cycle, plus a molecule of 2-phosphoglycolate, which undergoes reactions that release carbon dioxide and use ATP. Therefore, because photorespiration is releasing carbon dioxide instead of fixing it, and using ATP instead of generating it, we often think about photorespiration as doing the reverse of photosynthesis. As you might imagine, this is very costly for plants. Let's look at the conditions under which photorespiration is common. If we look at a cross-section of a leaf, we would see that inside the leaf there are many air spaces. These air spaces allow carbon dioxide to diffuse into the mesophyll cells and oxygen and water to diffuse out. Once the oxygen molecules are outside the cell, they fairly rapidly diffuse out of the leaf through holes called stomata. The stomata are surrounded by specialized cells called guard cells, and these cells can cause the stomata to open or close depending on light and depending on whether the leaf has enough water or not. So let's look at what happens on a dry, sunny day. When sun hits the mesophyll cells, photosynthesis is rapid, using up carbon dioxide within the leaf and producing oxygen and water. As the plant uses or loses water, the guard cells may close the stomata to reduce the loss of water. Once this happens, the oxygen that's produced by the photosynthesizing cells becomes more and more concentrated in the extracellular space, and carbon dioxide can't get in. This high concentration of oxygen and low concentration of carbon dioxide in the cells causes photorespiration to happen. Photorespiration is therefore a particular problem for plants in hot, dry, sunny conditions. Photorespiration was probably not much of an issue in the early evolution of plants because at that time the atmosphere contained very little oxygen. This inefficiency caused by high concentrations of oxygen is a relatively recent phenomenon, and photorespiration is therefore probably a relic of past evolution. Photosynthesis we've been looking at is generally called C3 photosynthesis because the carbon in carbon dioxide is attached to what quickly becomes a 3-carbon molecule. Some plants, however, have evolved a way to deal with photorespiration that uses a 4-carbon molecule. In plants that use C4 photosynthesis, there's a special arrangement of cells called Kranz anatomy. In these plants, mesophyll cells are still in contact with the air of the intercellular space. However, in these cells, a different enzyme called PEP carboxylase is used instead of Rubisco to bind the carbon in the carbon dioxide onto a molecule called PEP. The resulting molecule is a 4-carbon compound, and that compound is passed onto the bundle sheath cells when it's broken down to release carbon dioxide. Because of the specifics of the Kranz anatomy, the bundle sheath cells don't contact the air of the intercellular space. Therefore, no oxygen diffuses from that space into the bundle sheath cells. The concentration of carbon dioxide remains high, as it's essentially delivered directly into the cell from the adjacent mesophyll cells through plasmodesmata. The cycle occurs. The Calvin cycle occurs within the bundle sheath cells using rubisco. Photorespiration isn't a problem here because the carbon dioxide levels remain high and the oxygen levels low. We can therefore see that in C4 plants, the initial fixation of carbon and the Calvin cycle occur in different cells, separated in space. There's another group of plants called the Crassulaceae that use a similar mechanism to prevent photorespiration. This mechanism is called Crassulacean acid metabolism, or CAM. These plants are generally succulents and live in dry, arid climates. For them, water is very much in short supply, and so they avoid water loss by closing their stomata during the day when the air is drier and hotter, and water loss is a big problem. Instead of having their stomata open during the day, they open them during the night, and during that time they fix carbon dioxide into 4 carbon molecules. Then, during the day, their stomata are closed, and they use those stored C4 molecules to generate carbon dioxide, which is used in the Calvin cycle. They avoid photorespiration because the C4 molecule provides high concentrations of carbon dioxide to bind to rubisco. So you can see that both C4 and CAM plants accomplish the same thing, but in very different ways. In C4 plants, the carbon fixation and Calvin cycle are separated into 2 different cells. In CAM photosynthesis, carbon fixation and the Calvin cycle are separated in time, with the carbon fixation happening at night when carbon dioxide can be brought into the leaves without excessive loss of water.